Hello, everyone. My name is Victor Menezes, and I'll present my work on the assessment of focused ultrasound-mediated delivery of chemotherapy and cytotoxic activity in brain tumors with cancer-soluble biomarkers. Liquid biopsy uses blood sampling for tumor characterization. However, a major obstacle to characterize brain tumors using liquid biopsy is posed by the blood-brain barrier, also known as the BBB, which hinders the blood flow between the tumor environment and the circulatory system. As you may see on this image on the left, different kinds of tumors are already assessed using the liquid biopsy technique. However, it does not include brain tumors. Previous work has shown that transcranial microbubble-guided focused ultrasound can promote the disruption of the BBB. Therefore, combining liquid biopsy and microbubble-guided focused ultrasound creates new opportunities to assess FUS-targeted drug delivery and monitor the response to therapy. In our study, we hypothesized that by collecting blood samples at different time points, we would be able to assess this FUS treatment better. So, in our assessment, we include the combination of FUS system and liquid biopsy, and it would include three major parts. The first one is the tracking of tumor growth and tumor regression. As the tumor grows, we expect an increase in biomarker secretion due to cell activity and replication. As the tumor regresses, we expect an opposite trend, where the secretion rate decreases. Second, confirmation of BBB opening. We hypothesize that there will be a sharp increase of biomarkers when comparing pre- and post-treatment samples. Third, effectiveness of drug delivery, where by assessing different biomarkers, we may be able to observe the activity of live and dead cells when comparing pre- and post-treatment samples. And this post-treatment sample may include a longer time point than immediately after the treatment. In our study, gossalyciferase has been chosen as our proof of concept molecule because it helps us to ensure that the signal comes from the inoculated tumor and its secretion increases with cell activity, replication, and proliferation. Plus, we can assess this molecule by two methods, the luminescence signal and DNA processing. In order to start our study, we started with in vitro, where we selected certain drugs and treated cells with each of them. And what we observed is, depending on the drug that we treat, the lower will be the cell viability of that specific group. Next, we observed the luminescence signal from the respective groups. And it is interesting that the luminescence signal will follow the same trend as cell viability, indicating that cell viability and luminescence are directly related. Third, we assessed the cell-free DNA that was present in the same samples. In this third assessment, what is interesting is that there is no linear pattern, and we see that there is a sharp increase between control group and our doxorubicin group. However, temozolomide and carboplatin don't follow the same trend. So our question is, why is the doxorubicin group such an outlier, and why does it not follow the same trend as other drugs? So we did a literature review with the different type of cell death morphologies and observed the specific types that will lead to membrane rupture, because membrane rupture is directly related to the secretion of certain biomarkers. So we selected which were the different cell death types that lead to membrane rupture and correlated all of them with the drugs that we used for the treatments. You can see that carboplatin and temozolomide lead to cell death morphologies that don't lead to membrane rupture. However, the case is different for doxorubicin, which overlaps with many different kinds of cell death morphologies that will lead to membrane rupture. Once done with our in vitro work, we moved on to in vivo, where we inoculated tumors into different animals and separated them between FUS treatment and no FUS treatment. You can see that the tumor size was very consistent among groups. And when we compare a pre and post-treatment sample, we see a sharp increase in the secretion of gossaluciferase for the FUS treated group. However, for the no FUS treated group, we don't see much change, which is an indication that by disrupting the BBB, we can see a higher flow of this biomarker into the circulatory system. Next, we broke down those groups when splitting them between no treatment group, a drug-only group, an FUS treatment-only group, and FUS plus drug, the combination of both techniques. When we observe and analyze the drug-only group, it has a very similar effect to the no treatment group. When we observe the FUS-only group, we see a sharp increase in the secretion of biomarkers, which is an indication that by disrupting the BBB, we would see an increase, as it has been shown in the previous graph. However, when we observe the data for our FUS plus drug for 24 hours post-treatment, it's not the same case. We see an opposite trend. Even though both groups were treated with the FUS, the drug caused a sharp decrease in the secretion of gossaluciferase, which is an indication that the cell activity has been lowered due to the enhanced drug delivery. I would like to acknowledge NIH for sponsoring our research, our lab with Dr. Arvanitis, and feel free to reach out with any questions that you may have. Thank you.